All right, welcome back. I hope you're ready to go again. All right, so let's talk about a few examples. And this is from the same review article that we were talking about before from Lindsley and colleagues. And this is looking at a particular glutamate receptor with two different allosteric modulators that they call compound 1 and compound 2. And you see for compound 1 why it's called compound 1 as opposed to its IUPAC nomenclature. So we're looking at two different downstream responses from this receptor. So the first is calcium signaling, and the second is ERK activation. And so you can see that with respect to calcium signaling, that if you add this compound here, compound 2, it actually shifts the curve to the left. So remember that we talked about the different types of modulation. So this would be affinity modulation. So it increases the apparent affinity of the orthosteric ligand, the DHPG, shifting the curve to the left, but it doesn't change the efficacy, right? They end up at the same, if you have a saturating concentration of DHPG and you add a saturating concentration of this allosteric modulator, they end up at the same place. So it doesn't affect the efficacy, it only affects the potency of the orthosteric ligand. So let's look at what happens with the other downstream readout. So again, the same orthosteric ligand. And in this case, we're looking at ERK activation. So you can see that you get a small increase with just the CPPH alone, without the orthosteric ligand. So what that says is that there's partial agonism, but you don't see the same efficacy. So it's acting just like a partial agonist does. It stimulates the response by itself, but it limits the total efficacy. So even in the same allosteric ligand is having two different effects on two different readouts. So I guess you could consider this to be a biased allosteric modulation. Now this is an interesting set of tables, and the reason it's interesting is because it's a listing of different known allosteric modulators. And the reason it's interesting is because many of these are molecules that are also orthosteric ligands for other receptors. So for example, cannabidiol is actually an allosteric modulator of the opioid receptors. You can see aspirin and sodium salicylate are allosteric modulators of one of the endothelin receptors. So again, there are lots of pockets on lots of proteins, and so oftentimes we assume that each drug only binds one site, but that's seldom true. And so oftentimes the effects of drugs that we think may be due to it binding orthosteric site may actually be due to it binding a completely different protein in an allosteric site. Unfortunately, we don't have currently good ways to predict what would be a good allosteric binding site. And so for example, let's say if we have a receptor and we know what the orthosteric site is, we can do molecular docking and we can potentially identify molecules that bind to that site that might be agonists or antagonists. We don't have the ability to do that with allosteric modulators. We can't just look at a protein and say, oh, that would be a good potential allosteric binding site. We're just not at that level yet in terms of predicting allosteric modulators, but certainly a lot of people are working to get there. All right, here are some examples, and the point is not for you to memorize what the structures are. It's really to show you that a lot of these molecules have somewhat similar structures in the sense that they tend to be rich in rings and planar, and so in general hydrophobic. So it's not always true, but in a lot of cases, allosteric modulators interact primarily through hydrophobic interactions and bind to hydrophobic pockets in molecules. That's a gross generalization. It's probably wrong in many cases, but if you look at these molecules, you see that there are sort of standard themes in allosteric modulators, at least ones that have been discovered so far. All right, as another example of allosteric regulation, let's think a little bit about protein kinases. And so the point here is to expand our thinking a little bit beyond drug binding when we talk about what allosteric means. So in a more general sense, if all we're saying when we're talking about allosteric modulation is changing the conformation of the receptor, then it doesn't necessarily have to be a drug binding. So this would be an example of drug binding. But of course, many proteins are susceptible to modifications like phosphorylation. So that can shift the molecule from the T to the R state, for example. Some molecules, like photorhodopsin, for example, are sensitive to light. So when they absorb a photon, they undergo a conformational change. So you can think about all of these as allosteric modulators and in the same general theoretical framework. Now, these scary colored curves on the left here are Gibbs free energy traces. So if you took chemistry, you probably learned about activation energy and energy states. And so, of course, proteins want to be in their lowest energy state, and those are in the bottom of these little wells here. And so the reason we have different colors is these are the protein in different forms. So the apo form basically means nothing is bound. So it's the free kinase with no ligand bound. And so that's represented by the blue line here. And so if you follow this blue line, what it shows is the so-called open conformation has the lowest free energy, or another way of saying it is the most stable form of the enzyme thermodynamically. So what that means is most of the APO kinase is going to be an open conformation, which happens to be inactive as a protein kinase. If you titrate in ATP, so now you can see ATP is bound to this kinase. Now it changes this energy profile. So now this intermediate conformation is the most stable. And so most of the enzyme is going to be in that conformation. So it's not in the closed active conformation yet. It's still inactive. Likewise, if you bind the substrate, so these are two different binding sites. So ATP is binding up here, substrate is binding down here. You can see that also the lowest energy state for the substrate-bound kinase is also the intermediate conformation. Now, if you bind both at the same time, if you have the ATP bound and you have the substrate bound, and so it's a little hard to see here, but here's the ATP here, here's the substrate here, and this makes perfect sense because you want to catalyze the transfer of the phosphate from the protein, from the ATP to the protein. They have to be in close apposition. And now if you look at the energy profile in yellow here, you see that by far the lowest energy is in the closed conformation, which is the active conformation. So this is really a beautiful example of how binding of both of the substrates, the ATP as well as the protein that gets phosphorylated together, convert the enzyme into a conformation that's active. So in other words, another way to state that is that the enzyme is only active when it has both of its substrates bound. So here's some examples, theoretical examples of different kinds of inhibitors of protein kinases. So again, this is based on the idea that really the kinase can be in an active or inactive conformation. So you can inhibit the kinase, for example, by designing a molecule that binds where ATP binds. So the enzyme thinks that it's got ATP bound, but it's an antagonist, so it can't transfer ATP, so that blocks the activity of the enzyme. And that's the way most of our ATP competitive kinase inhibitors work, although there are allosteric inhibitors now that are coming on the market as well. So anyway, so this is an orthosteric binding site. This is direct competition between the inhibitor and ATP. So this is an example of an allosteric inhibitor that binds a completely different site. So it's not binding to the ATP binding site, it's binding way over here. But you can see that there's a conformational change. It actually changes the ATP binding site so that it can't bind ATP anymore. So it's not directly blocking ATP, but it's indirectly through this conformational change blocking ATP binding. Now in this case, similar idea, but instead of blocking the ATP binding site, it's changing the conformation so that the protein substrate can no longer be bound. So the main point of this is the idea that there are many ways that allosteric modulators can work, directly or indirectly. Now here are some examples of orthosteric as well as allosteric binding of various different inhibitors. So for example, gefitinib is shown in red here, so it binds to the epidermal growth factor receptor, and it's binding right in the ATP binding site. This is an example of an inhibitor, this PD318088, which is an allosteric inhibitor. So you can see the ATP is binding adjacent to it, and this inhibitor is binding at a different pocket. So it's clearly allosteric, because these molecules, in general, if they bind to the same site, they're going to compete with each other. So allosteric modulators don't compete with orthosteric site in general. Although we'll see some examples where it's more complicated, because it's possible you can have a molecule that would bind both sites that's big enough. The point here, though, is that these binding different sites, and by inducing the inactive conformation, this MEK inhibitor inactivates the enzyme, even though it doesn't block ATP binding. So this is an example of imatinib, which binds to the ATP binding pocket. Again, here's another example down here, CHEK1, allosteric kinase inhibitor. So the ATP binding pocket's up here, if you sort of compare these structures, but the inhibitor binds here. And then there are other cases, let's go back, other cases where the allosteric inhibitor binds to a completely different subunit. So for example, in AKT, which is a protein kinase that's involved in controlling cell survival, it's got what's called a pH domain, which is separate, it's attached to, but it's separate from the kinase domain. And it turns out that there are inhibitors that bind to the pH domain, and so they cause a conformational change in the pH domain that prevents activation of the kinase. So it doesn't necessarily even have to bind, so not only doesn't it have to bind the same site, there are cases where the allosteric inhibitor doesn't even bind to the same domain in the protein. All right, so here are some examples, again, of various different ATP competitive, as well as allosteric modulators. And so as you might imagine, because for ATP, you've got adenine, so you've got this big flat planar group, that many of the ATP competitive inhibitors have very similar planar groups that fit into that same binding pocket that normally the adenine ring fits into. But you can see that the allosteric modulators are somewhat more diverse. So anyway, the point is not to memorize these, it's just get a little bit of a feeling for what examples of some of these molecules look like. Now, this is a very important point for kinase inhibitors, is that if you have, so let's say you have a molecule that binds to the ATP binding site and to a kinase, and the kinase happens to be an allosterically regulated kinase in the sense that, let's say it's a dimer. So, and this is this is an example from real life. So it turns out that certain RAF kinase inhibitors, when they're used to treat cells, at low concentrations, they actually activate RAF, and at higher concentrations, they inhibit RAF. But this was a little confusing because when you develop an inhibitor, you want it to inhibit, not activate, and you want it to do the same thing at all concentrations. So what's the explanation for that behavior? Well, it turns out that RAF kinase is a homodimer, and it's regulated in the Monod-Weinmann-Changeau model. So when you have an inhibitor that binds to one subunit, if it changes the conformation of that subunit, it also changes the conformation of the other subunit. So it turns out that kinases can be in the in or the out conformation with respect to this DFG motif. So when the kinase is active, it's DFG out, and when it's inactive, it's DFG in. So this is one of the switches that happens during kinase activation. So what? Well, the reason this is important is because some of the first RAF kinase inhibitors bound to the ATP binding pocket, but they stabilized active conformation. Now, because this enzyme follows the Monod-Weinmann-Changeau model, what that means is that the other adjacent subunit of the dimer also has to be in the active conformation. So normally, perhaps, this enzyme both subunits would have been inactivated, but now as you titrate in low concentrations of your inhibitor, it binds to one side of the dimer. It doesn't bind both sides because the concentration is not high enough yet. But it binds one site in the dimer, and it causes a conformational change in both subunits, leading to active conformation. Now, of course, the inhibitor is bound to the ATP binding pocket. So the subunit that it's bound to is inactive because it can't bind ATP. But the adjacent subunit is not drug bound, and it can bind ATP, and now it's in active conformation. So what happens is you titrate in the compound, it activates the enzyme by converting it to the active conformation. As you titrate in higher concentrations of enzyme, now you bind both sites. You have high enough concentration of the inhibitor to saturate the enzyme. So now you can't bind any ATP at all, so the enzyme activity goes down. So you have this biphasic effect, where you get activation followed by inactivation as you ramp up the concentration. So the problem is, of course, is that when you take a drug, the levels go way up, and then they go way down, and you take another pill, let's say, and the concentrations go way up and go way down. So what that means is if you take this drug, you're going to be activating and inactivating your kinases at different times, which is not what you want to do. You want to inactivate it if you're trying to stop cancer, in this case. So what do? So what you'd like to have is you'd like to have a molecule that not only binds to the ATP binding pocket, but stabilizes the inactive conformation, so that when it binds to a subunit in the dimer, it not only inactivates that subunit, but it also inactivates the adjacent subunit because it converts it to the inactive conformation. So this is a lesson that was learned by trial and error when RAF inhibitors first went on the market, and there were lots of problems with resistance. This mechanism was discovered very quickly and published, and so now when people generate kinase inhibitors, they want to identify kinase inhibitors that stabilize the inactive DFGN conformation. In fact, they saw crystal structures such as these to look at what conformation these molecules are in. All right, so what do I want you to know from all of this? I'd like you to know what the advantages of allosteric drugs are compared to orthosteric drugs. So I give you a list of them, so you should be able to explain a few of those advantages. Again, all of this we talked about rests on the idea that most receptors, if not all receptors, exist in multiple conformational states. So we talked about the T and the R state. So that's the simplest case where you have two conformations and one's, let's say, active and one's inactive, or if it's channel, it's open or closed. We know that in real life, it's more complicated than that. So we talked about, for example, the G protein coupled receptors. And so we know that in those cases, there may be at least 3 different conformations. So of course, the receptor is in an inactive conformation largely when it's not bound to a ligand at all. But in some cases, you may have more than one response. And so you may have a biased agonist that can activate one response but not the other. So that implies you have at the very least 3 conformations, if not more. So the other thing is that we basically said that allosteric modulators can really do just about anything. And as of now, we can't really design molecules to do what we want. So we only discover them to be able to do what we want. But so basically, you have to screen for allosteric modulators. You can't predict one. You just have to screen through a lot of molecules, and if you're lucky, you'll find a few that do what you want them to do. And again, we said that they can work in lots of different ways. They can affect the binding of substrates for an enzyme indirectly by changing the conformation of the binding site. In some cases, they can change the affinity for the orthosteric ligand. In other cases, they change the efficacy of the orthosteric ligand. And in some cases, they can show agonism or antagonism on their own. So it seems a little confusing that allosteric modulators can do all these different things, but it's not confusing if you think of it in terms of the point here is that these different conformations in receptors, and so allosteric modulators are just changing those conformations. And so the idea is that you select the modulators that behave the way that you want. We talked a little bit about the idea that particularly with respect to G protein coupled receptors, they can activate multiple responses. And so there's a possibility of developing biased ligands to selectively stabilize specific receptor conformations. And again, this can be important because oftentimes you can find allosteric modulators that can distinguish or differentiate between family members, where it's very difficult to find an orthosteric ligand that can differentially affect family members because, again, the orthosteric binding site can be evolutionarily conserved in most cases. All right, and we used kinases as an idea that there are many types of allosteric control. So again, allosteric ligands is one type, but there's also different subunits bind to different substrates. So in the case of a kinase, there's a protein substrate that gets phosphorylated. There's ATP that provides the phosphate that's being transferred. So both of those binding to the enzyme cause conformational changes. In addition, it's oftentimes more complicated than that because kinases oftentimes are regulated by phosphorylation of various sites. And depending on the sites, those can be negative or positive modulators. And oftentimes they're also regulatory subunits that can be either positive or negative. I guess if they're negative, then it's called an inhibitor. But nonetheless, you can either have positive or negative regulatory subunits. All right, so this again goes back to the kinase example, the idea that allosteric modulators can work through many, many different modes. So they can affect substrate binding, ATP binding. It could even affect the phosphorylation. So for example, if you change the conformation so that the activating phosphorylation site no longer gets phosphorylated, that's going to indirectly inactivate the enzyme. In the case of the AKT inhibitor that binds the pH domain, it can influence protein-protein interaction between domains of the same protein. So many different ways that allosteric modulators can function. All right, so let's stop there.